Have you wondered how farmers know if their crops are getting sick before the crop even looks sick? That's exactly the problem my research is trying to solve. Hello everybody, my name is Philip Lawrence, a master's student in the dry bean breeding and computational biology program at the University of Guelph. My thesis is on the phenomics side of this Anthracnose project. You may be wondering, what is phenomics? Well, phenomics is essentially the measurement of how plants look and perform in the real world. My colleague Ashby is handling the genomics side. I'm focused on the early detection of anthracnose in dry beans using hyper and multispectral imaging, essentially cameras that can see wavelengths of light beyond what the human eye can detect. So this is including the use of multispectral drones flying over the field to identify early signs of disease in plants before any visible symptoms appear. The earlier we can detect it, the sooner breeders can act and develop more resistant varieties, which means more stable and reliable crops for Ontario growers. My colleague Ashby is working on the genomics side of the project, looking at the DNA of dry bean plants to identify which genes are responsible for anthracnose resistance. So while genomics is essential, a gene is basically just a blueprint. It can tell you what a plant is capable of, but not what you'll actually see in a real field. So while genomics gives us the blueprint, phenomics tells us how that blueprint is actually expressed in the real environment. So traditionally, that measurement has been done by people walking through fields, visually scoring plants one by one. The problem with this is that it's slow, labor intensive, and two people can look at the same plant and score it differently depending on lighting, experience, or even fatigue. So phenomics replaces this with imaging technology and data analysis. It's objective, repeatable, and can process far more plants more quickly, but the most important thing is that it can detect stress in a plant before visible symptoms even appear. Because by the time dark lesions show up on leaves, the disease has already been disrupting the plant for days, and yield and seed quality have already been negatively reduced. In a breeding program with hundreds of plant lines, the sooner you can spot which crop holds up better under disease, the faster you can make selections and move a breeding program forward. So this is where multispectral drones come in. When a plant is stressed from diseases like anthracnose, the way it reflects light changes. Chlorophyll breaks down, cell structure shift, and a drone's able to pick up these physiological changes before the human eye ever could. Beyond the disease detection, the drone also gives us a broader picture of how plants are actually growing and performing throughout the season. We can track things like how quickly a plant's canopy is developing, whether it's maintaining its vigor under stress, and its overall health, how it changes week by week, from early in the season all the way to harvest. And flying over the field gives us a map of every single crop in the plot and the fraction of the time that it would take to walk and score them manually. And what would normally take days can be done about in an hour or so. So the data we collect from drones isn't just useful for disease detection, it gives us measurable traits like how much light a plant is absorbing, how well it's growing, and how its health changes week by week throughout the season. When we compare that with genetic data, patterns begin to appear. So plants with certain genes consistently show up healthier under disease pressure, which confirms that those genes are actually working in a real field and not just on paper. So together, the field data and genetic data gives us a much clearer picture. So before we can trust what we're seeing in the field, we need to validate it in a controlled setting first. So fields are unpredictable. Temperature changes, rainfall varies, nutrient deficiencies can look similar to disease. There's just so many things happening. In a growth chamber, every plant gets the same amount of light, water, and nutrient. So any difference we see is strictly from the disease. So I had two groups, a control that received no disease and a treated group that was inoculated with anthracnose. Photographed every plant every single day for 21 days using a hyperspectral camera, which captures hundreds of wavelengths of light far beyond what we can see. So the goal was to find which wavelengths detect anthracnose the earliest, before any visible symptoms, and understand what disease progression actually looks like over time. This project is about building earlier and more objective tools for detecting anthracnose in dry beans. We're combining indoor experiments, drone imaging in the field, machine learning to create a system that can evaluate plants more efficiently than anything currently available to Ontario dry bean breeders. So my project has four main parts. One, which was to confirm which strain of Anthracnose affected Ontario driving fields in 2024. Two was to perform the indoor experiment to find the best wavelengths for early detection. Three was to apply those wavelengths across 195 diverse driving varieties to identify which spectral traits are linked to resistance. And four, validating all of it in a real field condition. So the end goal is that breeders can develop resistant varieties faster and growers can end up with more stable yield, better seed quality, and less reliance on fungicides. In terms of progress, we confirmed which strain of Anthracnose was responsible for the 2024 outbreak in Ontario fields. The growth chamber experiment is complete. I finished annotating about 1,260 unique images in QGIS. Next, I'll be moving on to the top view, which will be another 1,260 images. And once that's done, I'll be extracting the key wavelength results to select the best wavelengths for the field experiment. On the field side, we planted 195 diverse driving varieties at Elora and Woodstock in 2025. And we had drone imaging running through mid-August until harvest, which is mid-September. And that data is now being processed. And we're also getting ready to plant the second year of field trials in 2026. So there's still some analysis ahead, but the project is happily moving forward.